So thank you for asking me to speak, and I want to give you an overview of genetics of epilepsy. Genetics of epilepsy used to be a dirty word, and the reason for that is that historically genetics was one of the main drivers of stigma. You wouldn't talk about the fact that people had epilepsy in the family. It sadly was the subject of eugenic, the terrible era of eugenics many decades ago. But even when I started working in this area in the 1980s, there was often a considerable resistance to discuss genetics because of the stigma. And sadly, it's still true, particularly in the third world. People were prohibited or discouraged from partnering or marrying because of the history of epilepsy in the family. And unfortunately, genetics had a very negative impact on people with epilepsy. So the fact that I think we've come a long way to understanding it, and you've already heard from Dr. Jansen how important getting to the cause is, and that's where genetics has a lot to say. So I'm a neurologist. I'm an adult neurologist, although I have been involved with treating many adolescents and children. And as doctors, we are asked really to answer three main questions. One is what's wrong with the patient or the child, and we're pretty good at sorting that out now with all the modern methods of treatment. We're not perfect, but we're pretty good. We're asked, can you cure it? And sadly, still at this time, cures for epilepsy are pretty rare, but we're hoping that we're going to get there. And to re-emphasise what Flor just said, we're often asked what's caused it. And historically, we as the medical profession answered this extremely poorly. A consultation might go something like this: well, what caused the epilepsy in little Johnny? Was it the fact that the birth took too long, or the obstetrician didn't come in time? Was it the fact that he fell off the change table when he was 6 months? Was it the fact that he got a vaccination and then didn't seem to be the same afterwards? Now, all that is rubbish. But often as doctors, we would reinforce the misconceptions, which is actually very bad, because we didn't actually have very good ways of answering that question about what caused it. And what's become clear is that much of the causation of epilepsy is genetic, or at least contributes to it. And as Flor has actually already indicated, the biological understanding underpins new treatments, which are coming. They're not all, it's not there for everybody yet, but they're coming, and that's why genetics is so important. So the traditional view of the causation of epilepsy is like this. And Flor, it's already been shown, the modern classification, but we used to think that about a quarter of epilepsy had obvious acquired causes, and this is still true: head trauma, strokes, neoplasms, etc. But about 2/3 or 3/4 of epilepsy, it was unknown, it was called idiopathic, and genetics was acknowledged, but it was minimised, probably largely due to this stigma issue, and it certainly wasn't politically correct to go into this in many societies. And in the 1980s, the first attempts at molecular genetic characterisation began, and we've now got a lot of knowledge. You've all heard about the genetics revolution, the Human Genome Project, which I'll discuss briefly. And it's these monumental changes in our understanding of genetics that have led us to have the ability to make these advances. So what's happened? So there's been enormous advances in what we call monogenic gene discovery. That is, where a single gene is the major factor in causing the particular disease. And for those of you that did science in high school or in university, this is so-called Mendelian genetics, which we learned from Gregor Mendel, the monk who worked with peas. One gene has a big effect. Now, that's true of some human diseases, but in fact, more often than not, the diseases that run in families, common diseases like heart disease, hypertension, asthma, Alzheimer's disease and epilepsy are not monogenic for the most part, and these have been much more challenging. However, the monogenic causes are important and they become a part of everyday clinical practice in child neurology, and we can now look for these things and make the diagnoses and give the families closure. And as I've said already, this has been enabled by enormous leaps in genomics technology. And I think the major challenges in the next 5 years are the following. The first is developing precision therapies based on this, and you've heard about where we're at just at the moment. They're still not widely applicable, but there's great excitement, particularly about the genetic therapies, which we hope will develop as a result of the gene discoveries that are happening. Secondly, solving the remaining monogenic epilepsies, because when you don't get a diagnosis as a patient or a family, it's disappointing. And thirdly, understanding the complex non-monogenic epilepsy. So this applies to the common forms of epilepsy, particularly in adolescents and adults, the generalised epilepsies, many of the familial forms of focal epilepsy. These are, for the most part, non-monogenic, and we're only beginning to understand those. So our journey started with this remarkable family from Australia, and this is a pedigree or family tree of 5 generations, and individuals in black have the disorder, and this is a peculiar form of epilepsy called nocturnal frontal lobe epilepsy. And my colleague Ingrid Scheffer, who is a very prominent child neurologist in the area now, joined me to do a PhD in the early 90s. And we, together with colleagues in Adelaide and in Germany, figured out that this was due to a mutation in what's called an acetylcholine receptor. And this was the first gene for epilepsy, which we're very excited about. We thought we were going to solve a lot of problems. It turned out that this is still very rare, but it's true. But it was the paradigm that you could actually find the genes. And when we look more broadly at epilepsies, we can consider anybody with epilepsy lying on, I've got a pointer on that? Not really. There's a spectrum on the right where there are genetic factors, and on the left there are acquired factors. And as I said already, about 25% of patients with epilepsy have an obvious acquired factor, trauma, strokes, etc. And we used to think that the single gene epilepsies, as evidenced by those families where the thing runs through the family like a golden thread, were very rare. And in the middle, we had what we called epilepsies with polygenic inheritance, where multiple genes are believed to interact, and these are much harder to solve. What we've learnt is that the number of single gene epilepsies is much larger, and Dr. Jansen gave the figure that at least in childhood about a quarter of children with epilepsy have single gene epilepsies, and this has grown enormously, and this is due to predominantly what we call de novo mutations. So these are new mutations that have occurred in the mother's egg or the father's sperm, and it actually doesn't run in the family. Nobody's had it before, and the child is sadly the first one to have it because of this new mutation. But these are genes of high effect and are potential targets for precision therapy. So that was a big revelation when it first became obvious 10 to 20 years ago. More than that, you're all familiar with the fact that things kind of run in families. Heart disease runs in families, high blood pressure, asthma, and epilepsy. Not always with a family trees that have this sort of golden thread. And we talked relatively vaguely about genetic background, but we can now actually measure it. So this is a new exciting development where we will be able to individualise risk to patients. A big change happened a little over 20 years ago when the first draft of the Human Genome Project was announced. And that project cost $3 billion. And if you wanted to have your genome sequenced at that time in 2001, you could do it for a cool 100 million. And in fact, there was a little era of time in the early 2000s where it was kind of fashionable for the mega rich to get their genome sequenced because they could. You could buy a yacht or you could get your genome sequenced or whatever. And one of the fantastic advances has been how this technology has now escalated, and we can now get a genome sequence for a much more reasonable cost of under $1,000, or an exome, the coding sequence, for less than that, and it's become part of clinical practice. It's no more, in fact, it's probably cheaper than an MRI, and it's become routinely expensive. And to appreciate the speed of this and the amazing advances, there's a thing called Moore's Law, which is the doubling time of computer power. You know, when you buy a computer, it's usually about 3 or 4 times faster than the one you bought a few years ago, and 3 or 4 times faster than the one you bought a few years before that. So that is so-called Moore's Law. And the price of sequencing has dropped much, much faster than what we see in Moore's Law. So this is really spectacular advance of scientific technology. And those of us working in genetics have been the beneficiaries of that, and we hope that transfers to patients. Now, we're often asked how many epilepsy genes are there? And I used to think there might be a couple of dozen or maybe 100. But that's way undercalled, and we've recently done an inventory of this, and work led by one of our students, Karen Oliver, and this shows that there are now nearly 1,000 genes that are known to cause directly or raise the risk of epilepsy. And the vast majority are in the bottom, what we call the DEEs, the developmental and epileptic encephalopathies, kids with bad epilepsy and often intellectual disability. There's also a large group for what we call the MCDs, or malformations of cortical development. But the common epilepsies, genetic generalised epilepsy on the top, and the common focal epilepsies, the number discovered so far is relatively small, and that's because most of them are not single gene disorders. But we've got a large number of single gene disorders, particularly in the severe epilepsies of childhood, that have now given closure to the families. It wasn't due to the vaccine. It wasn't due to the fact that they fell off the change table. It wasn't due to the fact that they didn't get to the hospital fast enough for the birth. It was unfortunately programmed in the genes. And I think, and I hope you agree with me, that that's very important information, because many families carry the guilt. If I'd only done this or not done that, little Johnny or little Mary wouldn't have epilepsy, and that's for the most part not true. And I think as doctors, if we can reassure patients, families with that, that's actually quite important. Now, there's another remarkable genetic fact that's become much more important in epilepsy, and that's what's called mosaicism. You all know what mosaics are on your bathroom floor or whatever. And mosaicism is when there are mixed populations of cells, and it occurs in nature. You can see it in flowers, and it's been regarded as a curiosity in human disease. The back of the man here shows mosaicism for pigmentation. There are these lines that are not pigmented and the lines that are not. They're mixed. And what we've learnt now is that mosaicism is far more common than we ever thought. and it can be very hard to detect, but we've got new techniques now to detect very low-level mosaicism. And what's turned out, the way it's turned out is it's become clear that it's extremely important for some hitherto completely ill understood causes of epilepsy. For example, the one shown in the image on your right is what we call hemimegalencephaly. It's hard to show without a pointer. But here, literally half the brain is malformed, and these children would virtually never have a family history. But we've learnt that when the tissue is examined, there is a mosaic mutation in a very important pathway that is, again, potentially treatable, and mosaicism has now emerged as a really important cause of hitherto completely ill-understood epilepsies. A much more subtle version is in what we call focal cortical dysplasias, which are perhaps now the most important cause of epilepsies that undergo surgical treatment, and often very effectively. And these can have a very low rate proportion of these mosaic mutations. In the largest group of the epilepsies with polygenic inheritance, so these are the common forms of generalised epilepsy, absence epilepsy or juvenile myoclonic epilepsy, which you may have heard of. It's due to, we believe, multiple genes, not a single gene, and this is much harder to crack. These disorders are known to be genetic because if you look in twins, the twins have a very high concordance for it. That is, if one twin has it, the second twin is highly likely to have it. And that kind of screams at you as a researcher that this is genetic. However, it doesn't run through the family like a golden thread. Most often there'll be a single individual that has the disorder. Sometimes there'll be more than one individual, but not that often. And rarely there'll be a autosomal dominant where it does run through the family like a golden thread, but they're a minority. We just announced a study 3 days ago where we've had a really hard look at these complex genetics, and this has been done through the International League Against Epilepsy Consortium on Complex Epilepsy. This is something that no single person can do, and we had over 300 contributors. One of the central people was Giampiero Cavalieri from Dublin, and also Bobby Kuhleman from Utrecht, and those two and I led this, but we had a team of really able, largely young analysts and researchers who've banded together to really make a big advance in understanding this. And the key picture is this, and what this represents is a so-called Manhattan plot, because it looks like the skyscrapers in Manhattan. And what we're measuring is all of the 20,000, looking at potentially all of the 20,000 genes in the human genome. And by clever statistical techniques, when the plot gets above the red line, it's deemed to be significant and very likely to be true. And we're now seeing these peaks representing specific genes that are, we now have very strong evidence that this is the underlying basis of this complex epilepsy. And this has been a big nut to crack. And we hope that as these studies go on, we'll understand the pathways and again, be able to have new treatments. So what we've learnt compared to where we were is that much of that other, if you like, 2/3 of the cake, there have been a few new causes found, for example, autoimmune epilepsies, which are important, but probably not all that common. But much of the rest, we believe, has got a major genetic cause, and this is beginning to allow us to tell patients, what caused it, which I think is very important, and is opening the way to the precision therapies. So the role of genetics in epilepsy, I think, has been greatly underestimated, and part of that was this horrible social phenomenon of the stigma and associated with the terrible curse of eugenics from 100 years ago, but even more recently. And although we believe that there are major genetic causes, these large families are rare, although they were first way for us to get there. This de novo mutation phenomenon, the new mutations, is very important, as is mosaicism. But there isn't sort of a single gene for epilepsy. There are hundreds of genes and probably thousands now. And we know that the epileptic encephalopathies, these very nasty epilepsies of early childhood, are very heterogeneous. We're discovering new mechanisms. And as I've shown you with that Manhattan plot, we're beginning to understand the causes of the common epilepsies. So the impact of this is that precision diagnosis, that is telling people exactly what the cause is now a reality for many patients and families. We can achieve this in about 50% of people with the epileptic encephalopathies, and we're also beginning to understand it in the epilepsies of later onset in adult life. So that's what I wanted to tell you tonight, and I hope you found that useful.
Thank you very much. Now we have a couple of questions, so we will. There was a lot of questions actually came through during your presentation, so it's very difficult to narrow it down. And I suppose you spoke there about, how genome, testing has become more widely available. What is your opinion on the 23andMe and testing like that?
Okay, so 23andMe, which is the sort of mail-out genetic testing, is relatively inexpensive. It doesn't look directly at the genes. It measures so-called genetic markers, and with the profile, they're able to say that you're at particular risk of certain diseases. It's relatively inexact. It's also useful for determining your ethnicity. For example, there are different patterns seen in that in people from different regions of the world, but it's not a clinically it's not a means for clinical diagnosis. So you can't go and order that through your pharmacy and, make your diagnosis. You still need people like me. Okay, so it doesn't put us.
So you're not going to retire anytime soon?
No, I'm not going to retire yet. Yeah.
Okay, and then I suppose looking at the risk factors, and if a person's mother or father had bipolar, would this put them at a higher risk of developing epilepsy?
So a family history of bipolar disorder does not really put you at much higher risk of developing epilepsy, no.
Okay. There's some coming in last minute here. You'll see them if you're looking at the app. And is there a higher percentage of male versus female family members who have genetic forms of epilepsy?
No, look, it's pretty equally distributed between males and females. There are some of the so-called polygenic epilepsies that have a predominance in boys or girls, but it's a minor effect. And there are some very rare epilepsies that are sex specific, but for the most part, it's relatively equal between the genders.
Okay, and I'll combine the last two here that just popped up on my screen. And how would you, I suppose, support a person within Epilepsy Ireland? We support people in the community, but what would you say to a family who is afraid of passing on the gene or the potential implications of passing on a genetic mutation?
It's a good question. I mean, the first is that it's very dependent on the type of epilepsy. There are some genetic epilepsies that are, really very mild and self-limited, and in my view, should not alter your decision whether to have a family or not. There are some that are very nasty that run in families. One group that I've been working on are the so-called progressive myoclonus epilepsies. These are by and large, a very severe group of diseases. So if you can identify the specific variant, then you can counsel people about the risk. So where the gene is known, then you can give much more specific information. In certain cases, you could do prenatal diagnosis if that's acceptable, but that's not a reality at the moment for the majority of forms of epilepsy. But what you need is a good diagnosis and a good genetic counsellor, and they would help guide the patient and family through that sometimes complex decision making.
Thank you very much, Professor Berkovic.